Many of you have probably heard or have even been diagnosed with strep throat, but have you ever wondered what actually causes it? The answer: Streptococcus pyogenes, a bacterium also known as group A Streptococcus, which will be my topic for this video. This bacterium causes not only strep throat, but also a wide array of infections, including lethal ones. Looking at statistics, an estimated 700 million infections occur from the bacteria each year. And while the overall mortality rate for these infections is low at 0.1%, over 650,000 of the cases are severe and invasive, with these cases having a mortality rate of 25%, according to Aziz et al. 2010. So we ask ourselves, what makes it a successful pathogen? But before getting into this, I just want to clarify the term invasive in this context here. So, invasive infections are infections of bacteria into sterile parts of the body. And there are two types of invasive infections caused by Streptococcus pyogenes, and they are cellulitis and bacteremia. Cellulitis is the infection of subcutaneous tissue, which is the deepest layer of skin, and this usually causes inflammation of the skin and is associated with swelling. On the other hand, bacteremia is when the bacteria enter the bloodstream. When this occurs, symptoms such as nausea, fever, and vomiting appear. So coming back to the question of what makes Streptococcus pyogenes a successful pathogen, let's first take a look at its characteristics. So on this slide here, I've included a diagram of the bacteria to give you a better understanding of what the structure looks like. And I just want to highlight the main components that I will be talking about a little bit in this video, and those are the M proteins, the lipoteichoic acids, and the exotoxins. So really briefly, characteristics include that it is a facultative anaerobe, non-motile, and gram-positive. So if we were to test for its oxygen requirements, we would get the results of the bottom middle picture. And facultative anaerobes can grow with or without oxygen. And if we were to test for motility, we would get the results on the far right corner. And for gram-positive, we could do a gram stain, and that would result in the bacteria appearing purple. So it is important to recognize that these are gram-positive, as gram-positive bacteria do have thick layers of peptidoglycan and contain teichoic acids. And in this case of Streptococcus pyogenes, this includes lipoteichoic acids, which I will talk about a little bit more on the next slide. So, transmission and infection. I will start off by saying that this is exclusively a human pathogen, so it can't infect other species. Usually, transmission occurs through airborne droplets or direct contact with either contaminated surfaces or infected people. And once inside the body, it is hypothesized that there is a two-step adhesion process that allows the bacteria to attach to host cells. I will only briefly summarize this process, so just keep in mind that there is a lot more to it. So, during the first step, weak hydrophobic interactions between the bacteria and host cells are established, and this is initiated by the lipoteichoic acids. This allows the bacteria to overcome the electrostatic barrier to the host cell. After this, the second step can then occur, and this step involves specific proteins from the host cell binding to surface molecules from the bacteria, and these adhesions are irreversible. I'd also like to mention that Streptococcus pyogenes produce erythrogenic and cytolytic exotoxins, and these toxins induce inflammation by activating T cells and lyse host cells, respectively. But how do these bacteria avoid the immune defense mechanisms? One way is that they have phagocytosis resistance. This resistance occurs because the M protein found on the surface of the bacteria binds to the red blood cell membrane to avoid being engulfed. A second way they avoid being killed is by having their own killing mechanisms for neutrophils, which is a type of white blood cell. The bacteria kill neutrophils by secreting a specific enzyme. So, what can we do once the bacteria is inside our body? We can use antibiotics to treat Streptococcus pyogenes. Typically, penicillin and ampicillin are the favorable choices of antibiotics due to the bacteria's sensitivity to them. And I've included a chart over here that represents data taken from a Northwest Ethiopian hospital. And this research was done by KBD al. 2021. And this just shows how sensitive the bacteria are to penicillin and ampicillin compared to all the other choices of antibiotics. And on the right, I've included a picture of a antibiotic disc diffusion assay, and it shows that there is no bacterial growth occurring around the antibiotic. But how does the antibiotic actually work? Well, penicillin interferes with the production of peptidoglycan. It inhibits cross-linkage activity and prevents new cell wall formation. The bacteria ultimately die without their cell wall because they are not protected against the pressure from the extracellular environment. Now, even though there is treatment available, we want to reduce transmission as much as possible, especially since the bacteria can be lethal to some. And prevention can be done simply by hand washing and avoiding sharing personal items. Along my investigation of Streptococcus pyogenes, I came across a lot of research papers, and I have listed one here that I found particularly interesting by Marks et al. 2014, and they investigated the relationship between biofilm formation and growth of Streptococcus pyogenes. And what they actually did was go to a daycare center and took swabs of the entire place and even took swabs from the children's hands and found that you can actually culture bacteria from 22 to 37% of the children's hands. And this just goes to show how many bacteria we encounter on a daily basis and how important it is to try to reduce transmission. So hopefully, this has you thinking on how you can prevent transmission from occurring and by doing your part, and hopefully maybe encourage you to investigate your own bacteria to see how you can prevent that from spreading.